Today we learn about third generation polymerase chain reaction, that is digital PCR. Hello friends, I am Dr. Manushi Siddarth, and in this video I'll talk about an introduction of digital PCR, its fundamental concept plus its advantages over fluorescent based quantitative real-time PCR plus digital PCR applications. So stay tuned at the end, subscribe and like my channel. Polymerase chain reaction has been modified by three generations. As we know, the first generation of PCR relies on gel electrophoresis to analyze PCR products. But they have certain limitations, like they always challenged by low detection limit, having laborious operation, and have a single application, that is qualitative. Real-time quantitative PCR is the second generation of PCR. They can quantify the products with standard curves, but also show low tolerance to interfering substances. Digital PCR is the third generation of PCR that enables absolute quantification through partitioning the reaction. They are highly sensitive and accurate in molecular detection. Before going to talk about its fundamental working principle, I would like to discuss in short about its history. The concept of digital PCR was first described in 1992 by Sykes et al. They found that the combination of limiting dilution, endpoint PCR, and Poisson statistics could yield an absolute measure of nucleic acid concentration. In 1919, Vogelstein and Kinchler at Johns Hopkins University developed a method whereby a sample is diluted and partitioned to the extent that single template molecules can be amplified individually, each in a separate partition, and the product detected using fluorescent probes. From this method onwards, the term digital PCR was coined. However, it still needs laborious manual dilution of DNA samples, in which bias may arise. In 2003, Liu et al. Reformed the techniques by adding macrofluidic elements. To increase the accuracy of dilution and reducing the operation-derived error, micropumps and microvalves were introduced to partition liquids from DNA templates. But the use of microfluidic technique was still restricted due to high-cost chips. In 2011, droplet digital PCR was invented based on water-oil emulsion droplet technology. This technique featuring high throughput, automaticity, and cheapness. Since then, digital PCR was ushered into clinical work. Now come to the fundamental principle of the digital PCR. In the digital PCR, once the PCR reaction mixture is prepared, the PCR reaction mixture along with the template DNA partitioned into thousands of smaller compartments in which separate PCR reaction occurs in individual compartments. The mode of processing is similar to the real-time PCR. Once the fluorescent labeled probe binds to the DNA, it emits the light which is measured by the machine. The absolute quantification of target DNA is done using the Poisson distribution method, using the numbers of positive versus negative reaction done in each compartment. The fluorescence emitted by the compartment with no template is called a zero, or it is used as a baseline for the calculation, also called PCR negative reaction, which is described as a black color in figure. The amount of the fluorescent emitted by the compartment with a single template is called 1, which is also called PCR positive reaction, and it is described as a red color in figure. In the digital PCR, reaction mixture is divided into smaller microvolume reactions in such a way that only 1 or 0 target molecule may present per macrovolume reaction. In the final calculation, the total number of positive reactions having one template is equal to the total number of target DNA present in the original volume of a sample. The absolute concentration of total volume is calculated using this equation, Absolute concentration is equal to a total number of template DNA counted divided by total volume measured in each well or microvolume reaction.
So what are the advantages of using digital PCR? The main advantage of digital PCR is that they are highly sensitive and they can detect the difference in the gene expression of less than 30%. Only a single copy number variation can be distinguished accurately. In addition to this, it can identify the alleles having allelic frequencies less than 0.1%. There is no need to rely on reference or standard for quantification. They have higher reproducibility and improved multiplexing capacity. They have high tolerance capacity against PCR inhibitors.
According to the methods of separating reaction mixture, digital PCR can be classified into chip digital PCR and droplet digital PCR. The chip digital PCR depend on microfluidic technology and it involves the partitioning of reaction into nanoliter reaction chambers. Firstly, prepare the reaction mixture in a tube and this reaction mixture partitioned into individual chambers by microfluidic devices for PCR amplification. Each reaction chamber is approximately 0.6 nanoliter in volume. After cycles of reaction, fluorescence is detected with an imaging software or system and an inverted endoscopy. The reaction chambers with green color are assigned as positive reaction containing target template. Then the copy number of target sequence is calculated with imaging software. Droplet digital PCR creates macro reaction units with water in oil and macrofluidic technology. The nucleic acids are randomly subdivided into water in oil droplets that undergo PCR separately. Then, a two-color optical detection system is used to read the signal in each droplet. Through the process of water-oil emulsion technique, the nano-volume sized oil droplet is created for partitioning the reaction. The quantification of DNA using this technique is called droplet digital PCR. The droplet digital PCR generates more than 20,000 macrovolume partitions for absolute quantification. Higher the partitions, higher the accuracy. If the reaction is partitioned is larger number of microvolume reactions, the chances of having 0 or 1 template in macrovolume reaction compartment is higher. Thus, counting becomes more accurate. Thus, droplet digital PCR is advisable for rare allele or low frequency genotype detection. Here are some of the application of digital PCR. For rare mutation detection, digital PCR is very useful as we know that digital PCR can identify the alleles having allelic frequency less than 0.1%. So ultimately useful in cancer diagnosis. They also very much useful in absolute quantification, GMO detection, gene expression analysis, and also useful in non-invasive prenatal testing like chromosomal aneuploidy detection and autosomal recessive disorder. They also very much useful in quantification of next generation sequencing libraries. This is the reference of the literature where I used in my video. So thank you for listening. For upcoming videos, don't forget to subscribe and give thumbs up if you like this video. Stay tuned. Take care.